Good morning, all. Voice audible? Ayan Sharif. My voice is clear? Hello?
Yes, sir, it is clear.
Right. So now today topic is, last class we have discussed about the gene structure. In gene structure, one of the part remain, that is repetitive DNA. Are you able to see PPT slide?
Yes, sir.
Right. So repetitive DNA. What is repetitive DNA? The name itself given repetitive, that means the nucleotide series repeatedly formed. So that is called repetitive DNA. So here classified into, actually before repetitive, actually the gene mainly two types in human beings or any plants or other animals. Unique DNA sequence very less present, and highest repetitive DNA sequences. So actually this is present in human genome. So majorly the genome, two parts, unique DNA sequence, very less amount, and repetitive DNA sequence. Actually, this unique DNA sequence active, which is involved in transcription process and protein formation, the protein synthesis, or any other genetic material prepared by the unique DNA sequence. And rest of the gene part, that is called repetitive DNA sequence. If you see repetitive DNA sequence, it is again classified into two, that is tandem repeats or satellite DNA or satellite repeat, and interspersed or dispersed repeated DNA. Second one, dispersed repeated DNA. So first of all, I think the definition clear. Repetitive DNA sequence means here the specific nucleotide sequence formed repeatedly. That is called repetitive DNA sequence. The first, the tandem repeats. Tandem repeats means in tandem repeat, the nucleotide sequence formed one after another, repeatedly formed. For example, if you take the DNA, for example, this is DNA strand, and DNA strand. There are, for example, tell me any two nucleotides, any two adenine, guanine. So they are repeatedly formed. Adenine, guanine, adenine, guanine, adenine, guanine, adenine, guanine. Like this. So this repeated form called as the tandem. That means the in tandem repeats the repetition, the highest number when compared with the dispersed repeats. So this is tandem. And again tandem, the major one is tandem, another name satellites also. And in the satellites DNA, a mini satellite and microsatellite. The name itself given, mini satellite and microsatellite. Mini satellite. Actually, this repetition process, uh, somewhat, for example, for a mini satellite, if we take mini satellite, the repetition process, that is based on length. The micro very short repeated. The micro very short repeated than mini satellite. That is the difference. So anyway, in mini satellite and micro satellite, both repeated DNA, but the base station repetition, the repetition very short, frequently repeated with very short, that is called micro. Or somewhat the extent, that is called mini satellite. For example, here, so some sequences. It's the same, it can take same adenine, guanine repeated. But after another nucleotides common, another nucleotides are present. And again adenine, guanine. So this repetition very short. This repetition length, for example, mini satellite, the repetition of these nucleotides for every 10 to 20 nucleotides, 10 to 20 nucleotides. But micro, the 5 to 10 nucleotides between the gap, 5 to 10 nucleotides, micro satellites DNA repetition, mini 10 to 20 nucleotides repeated to DNA sequence. That is the difference between mini and satellite. And actually, in tandem repeats, the mainly present in heterochromatin. Do you know heterochromatin, euchromatin? Hello? Heterochromatin, euchromatin. Heterochromatin. What is heterochromatin? What is euchromatin? Where they are? Already we discussed in genome structure, chromosome morphological structure, chromonema, chromatids, chromomeres. There we discuss very briefly about heterochromatin and euchromatin. Heterochrome, when you stained the chemicals, it received dark stain. That is called heterochromatin. If receive light stain, that is called euchromatin, right? And this tandem repeat mainly present, this repetition mainly present at centromere region. Do you know where are the centromere? Where are the telomere? Where is that? Hello? Where are they? Where is telomere part? Rani, Hayan Sharif.
Particular
Where is first centromere?
nucleus, sir.
First of all, centromere is the part of
Part of
did
DNA.
not you attend the chromosome class? Did not you attend?
No, sir. Sorry, I did not attend the class. Today is my first.
Centromere is the part of chromosome which is located the center region. If you take chromosome structure,
Yes, sir. I forgot. Real. Yes, sir.
the centromere, and these are the two. It
Uh,
attached the two chromatids like this arrangement, and here somewhat knob-like structure present. That is in knob there SAT chromosome present, and the dispolar, the polar end of this chromosome structure. This is called telomere. This is called telomere. This is called SAT chromosome. This is centromere. So in chromosome, in centromere region, in telomere region, in SAT region, they are mainly present repetitive DNA sequence. And so actually, when compared with unique sequence, repeated DNA sequence high. In actually the tandem repeat in genome entered in genome 10 to 15%, and remaining percent of the repetitive is dispersed repetitive DNA. Dispersed repetitive DNA means not continuously formed. This repetition not continuous formed. The now and there this repetition occur. That is called dispersed or interspersed repetition. This dispersed somewhat it is involved in protein synthesis or some types of RNAs, formation synthesization of the some types of RNA. But tandem repeats, there is no gene. In tandem repeat DNA, there is no gene. It is not involved in any transcription process, any translation process, any protein synthesis. They are simply called as a junk DNA. They are simply called as junk. I will tell you, even it is junk, the some of the importance also. I will tell you later. First, remember these are the junk, and another next dispersed is this repetition present, but it is not repeatedly formed. So now when there somewhere the randomly this repetition occurs. So this is called dispersed repeat. And dispersed repeats, the major one is transposons, tDNAs, and retrogenes. In this major two, of course, in our book not given about I think transposons, but remember in dispersed interpersonal transposons and retroposons or retrogenes are there. The first, let's see transposons. The transposons means.
Sir, we cannot able to see the full slide.
Just wait. Now clear?
No,
Are you
sir.
able to see eraser? I'm erasing some of the lines.
Yes, sir, that we can able to see, but below part, whatever you're explaining now, sir, it is not visible. Up to transpose or tDNAs, up to there only we can able to see the slide.
So in repetitive DNA charts, tandem actually chart visible or not?
Full chart is not visible, sir. Only half chart only visible.
After then until
Until
can you
this.
able to see can you see tandem repeats, dispersed repeats,
Yes, sir, that we can able to see.
and
There,
tandem types?
that also we can able to see, sir. After that, whatever the types are there, that is not that are not visible.
That mean actually tandem repeats type, mini satellite and micro satellites, which are visible or not? In tandem, see one side. Are you able to see the pen marker? Able to see pen?
Yes, sir.
Able to see pen satellite DNA? Satellite DNA, is it visible or not?
No, sir, that is not visible.
Only tandem repeats visible, right?
Yes, sir.
Okay. In tandem repeats, remember mini or micro. Mini or micro, major two. Dispersed clear, interspersed dispersed repeats.
Clear, sir.
In dispersed repeats, transposons.
That also clear, sir. Transposons,
Is it
parallel.
visible? Transposons visible, parallel, actual parallel after the transposons clear.
Yes, sir. Clear, sir. That also clear.
Retro pseudogenes clear.
Yes, sir. Clear.
Right. Now let me explain about transposons. In transposons also repetitive DNA, but this repetition cut and paste. Cut and paste means, for example, here this is the DNA sequences. Some DNA sequence, thymine, adenine, cytosine, and here some other DNA sequences are there. But this, and again TAC, for so. In this repetition, actually this repetition not continuous. TAC, enough, and some other nucleotides between this, more than 100 nucleotides are there. Below the 100 nucleotides, that should be tandem repeats. And in tandem repeats, mini satellites, almost 20 nucleotide between repetition. In microsatellites, very less, 10, 5 to 10 nucleotides in between, right? So in this, what happened in transposons, this repetition, one of the repetitive DNA cut here and paste somewhere, So this is called repetition DNA, transposons repetition DNA. And another one, retroposons. The same retroposons also same like a transposon, but here the difference is transposon cut and paste. Retroposon genes copy and paste. Are you clear about this too? Hello? In dispersed repeats, transposons cut and paste. Retroposons, they are copy and paste. Hello?
Yes, sir. Clear, sir.
Clear. Right. So this transposons, another name called as jumping genes, because they are jump from one place to the another place. And in this, another type, actually it is not visible, but I will write here. In transposons, two types: line, another one is sign. Simple. Line means long interspersed nucleotide sequence. Long interspersed nucleotide sequence. Sign means short interspersed nucleotide sequence. Short interspersed nucleotide sequence. So this is two types. And name itself given long interspersed means this interspersed, for example, in this interspersed, this region, this type of nucleotides cut here or copy here and paste somewhere long distance, that is long. And if we take the short, cut here and copy here and paste very closely. So this is called short interpretations. So this is about repetitive DNA. Is it clear about repetitive DNA or any doubt in this? Hello?
Once you explain about paralogs, no, sir.
Actually, parallax is not important here. That's why it's no need to explain. There is no point to explain about tandem parallax and
Okay, sir.
dispersed parallax also.
Okay, sir.
Right. So once again, actually, this dispersed repeat, another name moderately repeat. In our book given, it is called as moderately repeat and highly repetitive DNA. Don't get confused these words. In page number 66, given clearly unique DNA sequence. One is unique DNA sequence, and second one is in repetitive DNA, two types repetitive DNA. What is two types? Moderately repetitive DNA and highly repetitive DNA. But in chart given in repetitive tandem. Tandem means highly repetitive DNA. And the second one, dispersed. Dispersed means moderately repetitive DNA. That is the difference. Dispersed, another name is moderately repetitive DNA. Right? Actually, this high repetitive or tandem not involved in protein synthesis, and which are present in telomere, centromere, satellite DNA, and this do not present any protein. Of course, it is not involved in protein synthesis. Even we called as junk DNA. They are mainly involved in chromosome organization, genome organization, like the binding of book, how the pages are binding in a book with help of the material. So like that in tandem repeats and highly repetitives involved in like a binding book, and mainly the involved in organization of genome. That is the importance of tandem repeat. And if we take the transposons, and that may moderately repeat involved in different type of RNA synthesis and protein synthesis. But due to this repetition, some diseases, some disorders occurs in human beings due to the repetition, due to the jumping genes. Uh, for example, majorly due to this, some mental disorders occurs in human beings. that is one of the default of this jumping genes, and the majorly these jumping genes the mostly importance in formation of ribosomal DNA. Ribosomal DNA, it is involved in ribosomal DNA. Only this is the only one function, formation of ribosomal DNA, right? So this is about repetitive DNA, right? Is it clear? Hello? Unique DNA sequence, that is unique for every individual. For this repetitive DNA, this is conserved DNA sequence. It is come from the generation to generation. In fingerprinting and DNA test, any DNA test, this repetitive DNA mainly involved. Especially in this mini satellite and microsat, based on the mini and microsatellites, DNA fingerprinting recognized. DNA fingerprinting recognized. Detecting fingerprinting recognized. Is it clear? Hello?
Clear.
Right. So this repetitive DNA also important in short answer. Okay. Genome structure clear. Gene structure clear in that is page number. The last class we completed this eukaryotic gene structure DNA level. In eukaryotic gene structure, we completed about UTR and alternative start and stop codons and overlapping genes, alternative splicing, gene family, and repetitive DNA. These are about genes. And another important one is in gene family, sorry, in gene structure. Another important, this is important in transcription and translation process. That is promoter, enhancer, and silencer. Promoter and enhancer and silencer. If you take, for example, just wait. I will explain during transcription, eukaryotic transcription. So next unit, the gene expression in eukaryotes. Gene expression in eukaryote. Fifth unit. This is very, very important. Gene expression eukaryote, very, very important. Let's take PPT slide. Are you able to see the slide? Hello? Is it visible to you?
Yes, sir.
Yes.
Eukaryote case types of RNA polymerases. Visible or not?
Yes, sir.
Types of RNA polymerase. Right. Right. Simple. Gene expression in eukaryotes. What expression the gene here? So, for example, this is the gene. This gene what express? Gene what express? Hello?
Sir.
Gene what express here? Gene responsible for?
Characters, sir.
Right. That's it. Characters.
Characters.
Characters, right. The character is expressed after formation of this related protein enzyme. Related protein enzyme. How these proteins formed? How the proteins are formed? What is protein synthesis? What are the subunits of protein synthesis?
Ribosome, sir.
That is ribosome, one of the cell organelle. But I am asking definition.
definition.
Or what is subunits? What are the subunits of protein?
Amino acids.
Right. How the amino acids are formed one by one. So anyway, okay, right. So group of amino acids together form as a protein. This group of amino acids attached on the RNA, mRNA strand, messenger RNA strand. Messenger RNA. After messenger RNA formation on the mRNA strand, one by one after another, amino acid attached, gained, and this amino acid chain formed. This group of amino acid chain called as protein. After formation protein, this protein enzyme react on the related characteristic substrate. Now the character expressed. Before that, how the mRNA formed? mRNA formed from the gene. mRNA formation from the gene. From the gene, mRNA strand prepared. This process called as transcription. Transcription. After transcription, mRNA strand prepared. Actually, this is in eukaryote, this mRNA not truly developed mRNA. First mRNA called hnRNA. After processing, this hnRNA developed as a mature RNA. The first gene, hnRNA, this hnRNA, after processing, developed into mRNA. After formation mRNA, on the mRNA strand, a number of amino acids gathered as a result formation of protein. Now this character is expressed. Now the character expressed. For example, let me give some brief introduction. For example, one of the in plant body, one of the character is flower producing, flower formation or flower producing. So the flower producing character gene is present. Gene is present, but this gene present from beginning or it is added in middle of the age? You tell me. Hello? Hello? Are you there?
Yes, sir, we are there.
I am asking simple question. I have taken one of the simple exam, that is production of flowers in plant. So for production of flower, that flower might be color or smell, whatever it may be, anyone that responsible one particular gene.
Yes,
This
sir.
gene is present. My simple doubt is this gene present from the beginning, or these genes introduced before the gene synthesized or introduced before the flower production?
No, sir. It is from the starting only.
Right, it is from the starting, but until it is inactive. When before producing, what happened from this gene synthesized through the transcription process, mRNA prepared. mRNA, like this is mRNA strand. On the mRNA strand, the amino acids are added. One amino acid. I will explain clearly, but let me give brief introduction. Say another amino acid, third amino acid, like this number of amino acid chain form. This group of amino acid together form as a protein. Now protein enzyme formed. After formation, this protein enzyme. Now this protein enzyme react on the flowering substrate. Now the flower expressed character. Which character expressed? Production of flower expressed. This is called, this total called central dogma. So in central dogma, gene or DNA transcription process, mRNA form, translation process, protein. So here are the two stages. One is transcription, another one is translation. Through the transcription, RNA strand prepared. and translation protein strand prepared. So this is called as a central dogma. This is called central dogma. So in this process, first let's complete about eukaryotic transcription. Now this is clear. Hello, transcription and translation clear or not? Hello,
Yes, sir.
transcription. What is transcription? What is transcription?
Transferring of DNA template to RNA.
Right. What is translation?
Translation.
Protein.
Right. Protein amino acid formation on the prepared mRNA. Amino acid formation or protein formation called as translation. Now in this part, first let me complete about transcription. This is very, very important for long answer and central dogma for short answer. In central dogma, if they are central dogma, just simply write about the two stages, transcription and translation. Already clearly given the simple flowchart in page number 88. Sorry, not 88. Page number where?
74, sir.
74, right? 74. Now let's see the eukaryotic transcription. Already, I think the definition clear. Transcription, what we prepared in transcription. In transcription, we prepared the mRNA strand, right? So let's take in transcription process. In transcription, one of the polymerase enzymes involved. Actually, if we compare with prokaryote, in prokaryote, actually only one polymerase, one type of polymerase enzyme involved in prokaryotic transcription. But in eukaryote, there are 5 types of RNA polymerases. 5 types. Are you able to see the marker? Hello? Hello?
Yes, sir. Yes, sir.
Right. So 5 types of RNA polymerases. The 5 types from 1 to 5. The RNA polymerase 1 and 2 and 3 are found in eukaryotes. All eukaryotes. While 4 and 5 are exclusively found in plants. Found in plants. Right. So first of all, RNA polymerase 1 function is the RNA polymerase 1 synthesizes 28S, 18S, and 5.8S rRNA, ribosomal RNA. One RNA polymerase 1 involved for synthesis of ribosomal
Sir,
RNA.
sir, PPT is not visible, sir, here. Just we are seeing the markers.
Only markers, just it.
Yes, sir.
That's what I am asking you.
In the local disk, it's there, sir, all the files.
Okay. Now visible or not?
Yes, sir, now it's visible.
Is it clear?
Yes,
Hello? Right. So first in transcription requirements. What are the requirements? The major requirement for transcription process, RNA polymerase enzyme. Already told you in prokaryote only one type of RNA polymerase. In eukaryote, 5 different types of RNA polymerase there. Let's discuss one by one. Here the 1 to 3 present all type of eukaryote, especially especially 4 and 5 exclusively found in plants. The first RNA polymerase 1, it is involved in synthesis of rRNA, ribosomal RNA, synthesis of rRNAs, ribosomal RNAs. In ribosomal RNAs, they are 28 years, 18 years, 5.8 years, they are all. And 2, RNA polymerase 2 synthesize mRNA, that means messenger RNA. Apart from the messenger RNA, it is also involved in snRNAs, spliceosome RNAs, snRNA or spliceosome RNAs. They are U1, U2, U4, U5. And last one, 3, RNA polymerase 3 synthesize 5S rRNA, 5S. Actually, these 3 involved in synthesis of tRNA. Apart from the tRNA, one subunit of 5S rRNA also synthesized. Actually, rRNA synthesized from which RNA polymerase? rRNA, ribosomal RNA from, hello?
Polymerase 1, sir.
1, actually 1. But 3 also involved only some subunits, 5S rRNA, very small subunit 5S rRNA prepared. And also the majorly it is involved for preparation of tRNA. And also U6 snRNA, RNA polymerase 4 and 5 synthesize siRNAs. Among 5 RNA polymerases, only polymerase 1 found in nucleolus. Nucleolus. The remaining are present in nucleoplasm. Do you know where is nucleoplasm? Where is nucleolus? If you take nucleus, this portion nucleolus, the rest of the part called as nucleoplasm. So only one prepared in nucleolus, only one present in nucleolus. The rest of the all present in nucleus or nucleoplasm, only are nucleoplasm. So this is about RNA polymerases enzyme. So here for transcription process, we required major mRNA. That's why we discussed about only which RNA, which enzyme, which RNA polymerase enzyme. mRNA preparation from which mRNA? 1 and 2 and 3.
2,
Two.
sir.
So let's discuss about RNA polymerase 2. So you don't forget about the concept of the transcription. What is the concept of transcription? Preparation of mRNA, right? For preparation mRNA from where? From where mRNA prepared?
From DNA.
Right. Actually, the structure of RNA polymerase not important, no need. Structure of RNA polymerase, this RNA polymerase large multi-subunits protein, this is no need. This is also no need. RNA polymerase 12 subunits, so 1 to 12 large subunit RBP1, RBP2. This is also no need. The promoter, I will explain about promoter sequences. Right. And here this is important one. For transcription require RNA polymerase 2. Okay. Apart from the RNA polymerase 2 requires another nuclear proteins. These are called general transcription factors. General transcription factors such as transcription factor 2D, A, B, F, E, H. These are the major transcription factors. I will explain one by one later. Functions of D, A, B, and F and E. Right. First of all, write down all this or take a picture, take a snap, because in our book, actually, whether it is given or not, where is transcription factor? Okay, right. Eukaryotic transcription, they're not detailed about RNA polymerase 1, RNA polymerase 2. You should write about RNA polymerase 2 and RNA polymerase requirement factors also. Right, until H. Right. In transcription process, 3 steps. One is initiation. Second one is elongation. Third one is termination. Already given in our book, page number 75. Process of transcription, 3 phases: transcription initiation, chain elongation, chain termination. These 3 clear? Hello? Hello?
Yes, sir.
Right. Actually, before the steps of transcription, we learnt what is transcription, what are requirement from requirement for transcription. What transcription means from the DNA mRNA strand prepared. For this requirement are major RNA polymerases. For preparation of mRNA, especially RNA polymerase 2. Only RNA polymerase 2 cannot perform the function without factors. So what are the factors for initiation of this transcription process? What are the factors? TF means transcription factor, 2 means RNA polymerase 2. Tell me, what are the factors? The factors are transcription factor D, A, B, and E, F, and H. They are the transcription factors. Without this factor, RNA polymerase enzyme cannot perform any function, any action. Remember it. Is it clear? Hello? Now let's see the steps of transcription. Initiation, elongation chain, termination of the chain. Which chain? mRNA chain. Right? Is it clear? Hello?
Yes, sir.
Right. Now, the major question is, actually long DNA strand, long gene is present in human beings. Where exactly transcription start? Before that, we should know the gene structure. For example, are you able to see marker? Hello?
Yes, sir.
Right. Please be careful. This is very, very important. For example, this is the DNA strand. In entire DNA, I would take only gene. For example, this is the one gene strand. Don't get confused between DNA and gene. I already told you what is the difference between DNA and strand. Gene is the part of DNA. Remember one word. Okay? Hello? Hello?
Yes, sir.
Right.
Sir.
Tell me. Any doubt? Right. So in this, actually this is the entire length of the total the different type of length. Here for where the transcription initiated. For the initiation, there is separate region present. That region called as, let me change color, promoter region. Promoter region. This blue color promoter region. In promoter region, there are specified sequences are there. Consequence sequences, specialized sequences there. I will explain later. And the promoter just close to the start site. Let's change color. Green. This is start point. Start point. Start point right side plus left side minus, and outside this is called as downstream. This is important because this repeatedly listen these words downstream and upstream. And also in your book also repeatedly appeared these words down and up. This is upstream. This is downstream. Right? So the blue color promoter. This is promoter. This promoter have the specialized consequences sequences are there. This sequence is called Tata box. This is very important. Tata box. Called as Tata box. So for the eukaryotic transcription, the regulated DNA sequence for the gene transcribed by each of the 3 RNA polymerases, but we have discussed only 2. Only RNA polymerase 2. Promoter, enhancer. Let me change color. Red. Enhancer. Enhancer at upstream. Present at upstream. So first promoter clear. Blue color region. Promoter is always found adjacent to the start point. Adjacent to start point. This is start point, adjacent to the start point. The promoters of genes RNA polymerase to contain 3 distinct regions, which are centered at site lying between 25, 30, minus 25 nitrogen base pair minus 30 of the upstream. This is upstream, already told you, upstream. And in this region, mainly the consequences sequences present. That is called Tata, TA. So Tata box. Or this another name is Goldberg, Agnes box. And another name is Goldberg, Agnes, because this sequences discovered by Goldberg, Agnes. That's why called as a Goldberg, Agnes box. Right? Like that, another consequences sequences also present in promoter such as cat box and GC box. But anyway, one important remember that is Tata box. Right? And next one is enhancer. Eukaryotic promoters also consist of site located 100 to 200 base pair upstream, 100 to 200 base pair upstream, which interact with proteins other than RNA polymerase and regulate activity of promoter. What is the function of enhancer? This enhancer regulate the promoter activity. So this site called as enhancement. And another one is silencer. Actually, this silencer, one of the regulatory gene, this silencer may be present at downstream and upstream. The function of silencer, this regulate, which is repress the gene expression. The silencer function repress the gene expression. Both the enhancer and silencer can function at great distance from the genes. They enhance and they repress one is enhance, one is repress. One is promote the promoter, one is repress. Repressor genes or silencer repress the promoter. What happen if the promoter repress? What happen if the promoter enhance? Tell me. What happen if the promoter enhance? What happen? Hello? Already told you this question, this topic is not an easy. Be careful. Tell me, I'm asking very simple question. First of all, in gene structure, promoter, silencer, enhancer, clear? Hello?
Yes, sir.
Right. My question is, if enhancer genes activate promoter box, what happen? What promote here? What enhance here? The enhancing the transcription process.
Yes, sir. Yes, sir.
Silencer gene, the stop the transcription process. The regulate. These are the regulate. One is enhance, one is regulate, one is repress. Is it clear? Hello?
Yes, sir.
Right, right. Now coming to the main topic, initiation. Let's see how the promoter initiated to transcription process. When enhancer activated, how they are transcribed. Let's see. First of all, what are the requirement for transcription?
Promoter, sir.
That is promoter site. So for promoter site, what are requirement? Promoter site is the part of DNA gene, but on the promoter site, which are requirement to synthesize transcription, to perform transcription. What are the requirement for transcription process? From the beginning, I told you. Ayyo. Are you able to see this diagram?
Yes, sir.
Right. What is this? This is this part. This one. This is RNA polymerase 2. Remember, RNA polymerase 2. RNA polymerase 2. RNA polymerase 2. First, what happened? Let's see. First, before RNA polymerase 2 attached to the promoter site, first of all, transcription factors attached to the promoter site. In this one, the first transcription factor 2 binded promoter site, transcription 2D binded promoter site. This is promoter Tata sequence. This is the promoter sequence, promoter site. And after that, actually TBB, transcription binding protein, bind first this one, next transcription binding protein. When transcription binding protein bind at the promoter site, automatically the DNA stands bent somewhat, bent down. bent down. Now the straight, after binding TBP, the bending shows the bending leg structure. Now the transcription factor 2D permit the association of transcription factor A and B. Now transcription factor 2B forms the so-called DB complex. After formation B, it is formation one of the B complex and RNA polymerase 2 associated to the promoter site. Now, after DNA transcription factor 2B attached to the promoter site, now RNA polymerase, this is RNA polymerase and RNA polymerase 2 associated to promote site. It is accomplished by transcription factor F associated with polymerase 2. Now after attachment TBP, D and B, RNA polymerase with the transcription factor F attached at promoter site to form the transcription complex. Now this is transcription complex. Now this transcription closed complex. That means now the completely close the promoter site. So that is called closed complex. Next, orderly addition of transcription factor E, transcription factor H and J helps in initiation factors. Right. So finally H and J. H joined later. So this order very, very important. Hello? Is it clear first? Initiation very complex. Be careful. Is it clear or not first? Hello?
Yes, sir.
If you have any doubt, any simple doubts, please ask me. It might be silly. Even the silly ask me because this is very complex. Clear? Hello? Hello?
Yes, sir.
Right.
Sir,
After
once in short you explain us, sir, initiation. Short.
Let me explain in some steps. First of all, you know promoter site. Hello?
Yes, sir.
Promoter is the part of DNA. Promoter. Before that, I have clearly told you the promoter sequence assists to the transcription process. Entire DNA, there is a special consequence sequence. The special consequence sequence region called as promoter. The consequence sequence also told you the consequence sequence are, what are the consequences? TA. That is consequence sequence, right? Before binding, actually, the promoter site binding with RNA polymerase 2, then only from the start point, mRNA prepared. Before start, the promoter site, the first transcription factors D attached. Next, transcription binding protein attached. Second, what happened when transcription binding protein attached? Then now the straight DNA downwards like cross, so as to easy transcription, so that the transcription easy when the DNA bent down. Now A now till now are alphabetical. B After B formation, next step, F, transcription factor F with 2 means polymerase 2.
Polymerase 2.
Right, this polymerase 2, actually, how the polymerase 2 reach the promoter site? Actually, this polymerase 2 cannot reach promoter site, cannot perform transcription process without these factors. Without these factors, what we discussing these factors, without these factors, there is no performance. So after 2, the next what happened in next steps? E H also associated with the RNA polymerase 3. Now the complex completed. Now this all called as, please be silent. Hello. Saujanya. Right. Now this is called as RNA polymerase 2 complex. Now this is closed the promoter site. Now this is called as closed complex. Is it clear now?
Yes, sir.
Any doubt?
No.
Right. This is initiation. What initiation here? The transcription initiation. Right. Now, closed 1, 2, 3. Next, elongation. So after attachment, transcription factor E and H, now initiation process started. Later elongation. Now initiation completed. Now this is closed complex. Later DNA unwinding to produce open complex. The open complex unwind. Right. Here also some elongation factors involved to transcription to mRNA strand elongation. What happened later, before elongation, unwinding DNA helicals. That means how the DNA, this spirally arranged, right? Hello? Actually, for our convenience, I have drawn like this. Or convince actually DNA I have drawn like a ladder shape. Actually, this DNA in this like spiral. So what in elongation, before elongation, what happened? This helix is unwind. Helices are unwind. Gradual helices are unwind. Now it is formed as a ladder shape. Helices are unwind and formed as a ladder shape. Let's see the elongation process. First of all, initiation clear. Initiation factors all clear.
Clear.
Right. Now let's see elongation. Right. Immediately after period of abortive initiation and synthesis of series of short transcript, the RNA polymerase escape the promoter. Now actually in initiation where the RNA polymerase exactly at promoter side. Now the promoter side, it move forward toward the downstream. Where is downstream? Don't get confused upstream and downstream. Downstream, forward or backward? Downstream. Okay, right. Forward direction.
Forward. Downstream.
Right. Exactly. Now this RNA move towards the downstream. So that means the promoter and enter elongation phase. When it is moved towards the downstream, automatically it is enter the elongation phase. Now the promoter escape involved in two steps. What happened during this elongation process? Actually, these two steps not seen in prokaryote. One step is ATP, adenosine triphosphate hydrolysis, and another one is phosphorylation. C-terminal RNA of polymerase 2. This is the part of RNA polymerase 2. Phosphorylation by the kinase activity of the transcription factor H. H for L capital గుర్తుంచుకోండి. H. The transition from the initiation to elongation involves shedding GTF and binding another site of transcription factors called elongation factors. Already told you, like initiation factors, here elongation factors also requirement. Among these factors, elongation factor L and TFPFB stimulates elongation. These two stimulate elongation, while TF2S stimulates the elongation as well as proofreading. Proofreading also. You don't get confused here. Simple. What are the two steps here? The first one, ATP hydrolysis, and another one is phosphorylation. That means simple phosphorylation means, for example, here in DNA T present. A, C. The first in mRNA, phosphorylated adenine, uracil, guanine. So the first, for example, adenine phosphorylated means, for example, the thymine phosphorylated with adenine. Now adenine phosphorylated uracil and guanine. Like this, new nucleotides are formed. Now this nucleotide formation continuation until reach the termination region. So this process called elongation. In this elongation first required for the formation of these nucleotides, mRNA nucleotides require ATP energy. This energy is utilized for the phosphorylation of CTD. That means C-terminal disphase of RNA polymerase 2. So in RNA polymerase 2, they are formation for synthesis of AUG. Now this sequence continuation. Like this, mRNA strand prepared. This strand goes continuation until reach termination period. For the elongation, actually first this kinase activity, that means phosphorylation kinase activity done by transcription factor H. After formation, one nucleotide of RNA. Now this is continuation elongation. It's keep continuation with help of the elongation factors. Those elongation factors are L and TEFB, which are stimulates the elongation, and S stimulates the elongation as a proofread. Proofread means, for example, here the TAC in DNA, somewhere GCATG. The proofreading means adenine, thymine, where it is, thymine, adenine. adenine instead of thymine, uracil formed in RNA, uracil. CG, uracil, CG, G, cytosine, guanine, adenine, uracil. So this sequence complementary to the DNA strand. And if any one nucleotide miss, it is not complementary. So here the function of transcription factor S, it ensure the all nucleotides, complementary nucleotides, whether formed or not. It ensure the complementary nucleotides in newly performing mRNA, the same complementary sequence formed or not. It is ensured by transcription factor 2S. So this is called proofreading. If any untoward nitrogen base is formed, this nitrogen bases untoward nitrogen bases removed by transcription factor 2S. This is called proofreading. Though any other junk material also present, this junk material also to be removed. So this is called proofreading. Now it is clear. Any doubt in elongation?
Clear, sir.
Right. So initiation clear and elongation also clear. Right?
CTD means, sir?
What?
CTD.
C-terminal. C-terminal of which is DNA part. Sorry, RNA polymerase part. C-terminal. Right. Now, you might have doubt. Actually, in this DNA, what are present along with the DNA? Already you know.
Proteins.
Right, histone proteins. Actually, the histone proteins are like a barrier. Like, for example, do you know nucleosome model?
Yes, sir.
And this is somewhere the nucleotides, histone proteins, DNA strand, histone protein, DNA strand, right? So now here the RNA polymerase, let's take, this is RNA polymerase. Actually, RNA polymerase move downward direction and synthesizing the complementary sequences on the downstream. Like mRNA strand goes preparing, continuous in preparing, but already you know the in this chromatin structure, apart from the DNA, histone proteins also present. Actually, these histone proteins, if the presence of histone protein, now RNA polymerase can move forward. Hello? If any histone proteins are present, apart from the DNA, the RNA polymerase moves easily. Can it move easily forward direction, downstream? Hello?
If histone proteins are there, it cannot.
Right. Actually, histone proteins stop the moving of RNA polymerase. These are the potential barriers to the presence of histones that that stops the elongation process. But prevent this, the factor called FACT facilitates chromatin transcription. Dismantle the histone octamer by removing H2A, H2B dimer. Now what happened? If the H2E, H2B, they are the separated. Now it is make it easy to synthesizing RNA, mRNA. Now thus the making this way the smooth for the enzyme to move forward. Right? So to remove this histone octamer, the FACT facilitates chromatin transcription. FACT, protein name FACT, facilitate chromatin transcription. React on histone and dismantle or remove the subunits of octamer, H2A, H2B dimer. If the H2A, H2B, A and B separate, there is no rigidity to the histone protein. That's why they are all the remaining histones also separated. Now after removing the histones, now only DNA present. Now the RNA polymerase will move forward direction. Is it clear? Hello?
Yes, sir.
Right. Right. Now what is the last step? What is the last step? Elongation
Termination.
completed, initiation, first initiated, right,
Termination.
termination. Now let's see how it terminate. Here the given diagram also RNA polymerase to move forward. Actually, these are the histone proteins. If histone proteins, H2A, H2B dimer separated with help of the fact, actually not given here. The fact, if fact attached here, automatically the 2A, 2B dimer separated. Now it is continuously move forward direction. Termination end. Termination two models, torpedo model and allosteric model. First of all, let's complete torpedo model. In our book also, that's very not given, I think. Elongation clear, elongation. Where is the elongation? Transcription initiation, right? Promoter class promise 2, okay. 2x, okay. D first, okay. Right, okay. Termination. Eukaryotic genes actually have no strong termination sequences like prokaryotes. Actually, in prokaryotes, there are row factor, the specialized sequence present, but in eukaryotic, no strong termination. The transition termination event occurs at multiple sites located at 1000 to 200 nucleotide downstream from the site that becomes the end of the 3 end. Actually, the mRNA start from 5 end it towards the 3 end. Right. Let's see this termination. Termination means end of the preparation of the mRNA strand. In termination first, let's see the torpedo model. In termination, transcription by RNA polymerase is not happened at specific sequence. There is no specific sequence. Acid RNA polymerase 2 often continues to synthesize RNA hundreds or even thousands of nucleotides. This keep continuing. When RNA polymerase transcribe the template DNA downstream, the poly A site, I will explain later. Poly A site is attacked by RNase. I will tell you after in the processing of mRNA, what is poly A, what is 5-methyl cap, what is placeuosome. I will explain again. Okay, right? What? Just wait. Right, RNase here, help of RNase. Where the poly repeated A sequence present, now it is attacked by RNase. That degrades RNA in 5 to 3 direction until the catch up to still transcribing polymerase from which RNA is being spewed. Separated. The RNase either pushes RNA polymerase forward or pulls remain the nascent RNA transcript means new RNAs, new RNA nucleotides from the enzyme, thus remaining the terminating transcription. This model is known as torpedo model. The simple in torpedo model, what happened? For example, DNA strand, the polyadenine tails present. If polyadenine tails present, automatically the mRNA strand become weak. Now here RNA is, now the RNA is RNA is attacked. RNA is attacked to the poly A. When RNA is attacked, no, RNA polymerase separated from the strand. RNA polymerase separated. With help of RNase, at the site of polyadenine, RNA polymerase separated. What happened if there is no RNA polymerase? There is no RNA polymerase. What happened? The transcription process go forward. Can transcription process go forward without RNA polymerase? Hello? Hello?
Sir.
Without RNA polymerase, transcription process move forward. Can it move?
No, sir.
No. That means the RNA is either pushes RNA polymerase forwarded or pulls remain of the nascent RNA. Here, don't get confused. The nascent RNA. Nascent RNA means what happened here. For example, this RNA polymerase present. This is DNA. Now, blue color mRNA. Nascent RNAs means this with help of this RNA polymerase. For example, adenine. Now, newly formed nucleotide from present uracil. One in DNA, cytosine present. These are newly nucleotides are formed. This until the nucleotide formation, the mRNA strand goes forward. These nucleotide sequences formed goes forward. But when RNase attack the polyadenine tail, where the polyadenine tail present, automatically RNA is attacked. After RNA is attacked, automatically this RNA polymerase enzyme separated, emitted out. This RNA polymerase enzyme emitted out. This RNA polymerase emitted out. Without RNA polymerase, there is no transcription. That's why it is terminated. So remember the terminated by RNase. Terminated by in this process, terminated by RNase. Right? Let's see another process. In another process, allosteric modulation. In what happened allosteric modulation? In allosteric model termination, there is another alternative model called allosteric model. The state terminate transcription occur through a conformational change. Conformational changes elongating polymerase. According to this model, immediately after transcribing poly A tail, RNA polymerase become less processive. That leads to spontaneous termination soon afterward. So after termination, RNA polymerase is dephosphorylated. Dephosphorylated means not phosphorylated to DNA, to mRNA, right? The release recycle initiated another round of transcription. The simple conformational changes. What happened? Conformational changes means here. Conformational changes in elongation
Means the structure of RNA polymerase is changed.
right, elongating what
The structure of the RNA polymerase might be modified or changed.
That's it. Actually, what happened poly A in torpedo, there is a RNA is attacked, but there is no RNA is attacked. Automatically, some of the structural changes occurs in mRNA. Due to the structural changes, this RNA polymerase not in active, less active. Now, automatically, so for example, let me give the simple diagram. What happened? Conformational changes. For example, this is the mRNA strand where the poly A present. This straight line formation loop-like appearance like this. Now, where here RNA polymerase present. Now, RNA polymerase move forward. Where the conformational changes, structural changes occur, like a loop formation. Can it move forward? Hello?
Sir.
Now can move forward. Now RNA polymerase enzyme move forward after
No, sir.
conformational changes like cannot move.
No.
And as a result, dephosphorylated. Dephosphorylated means actually always the phosphorylation formation, like for example, guanine present, automatically cytosine formed in mRNA, cytosine present, guanine present. This is synthesized continuously. But after formation, this conformation changes, dephosphorylated. No phosphorylation occurs between DNA and RNA. Without phosphorylation, there is no chain, no mRNA strand. That's why automatically the mRNA strand would be stopped. This is by changes, structural changes. The first one by with help of enzyme, RNase enzyme. First one, the simple with help of RNA enzyme. The both are attack the poly A tail, polyadenine tail. I will tell you how the formation poly A tail. The both formation poly A tail, but one is with help of the RNase enzyme attack. Another one is allosteric model by conformational changes. By conformational changes in DNA. So this is termination. Is it clear? Hello?
Yes, sir.
The simple in our book write about the allosteric and a turbid model. Two models enough. Now anyway, the final what happened? Final what happened here for DNA from the DNA strand somehow mRNA prepared. Let's draw a simple diagram. So this is DNA. Are you able to see diagram?
Yes, sir.
Right. So this is RNA polymerase move forward, where the poly A present. Now it is stop. Now stop. Now anyway, the RNA, mRNA, this is DNA. mRNA prepared. mRNA prepared. But this mRNA, this prepared mRNA not matured one. This not matured one. This not matured one. This is called as hnRNA from the DNA. Actually, first hnRNA formed. After processing, mature mRNA synthesized. In processing, first of all, do you know introns, exons? In DNA, gene, introns and exons also present. In DNA, introns and exons also present. In hnRNA, let's draw. This is red color and blue color, and again red color, like this. In the middle, blue color. These are formed. This is hnRNA. And fine, there is no protection. And here the poly A also not formed. So this is hnRNA, right? So now this is processed. During process, first of all, in first step, splices occur, spliceosome, splices occur. Second one is 5-methyl capping formation. Third one is poly A formation. Polymer formation. What is splicing? Splicing remove of the waste material introns. This blue color introns remove. Now all the active exons are gathered. After that, 5 cap formed at the 5 end region and poly A tail formed. 5 cap and poly A, these are the most important for the attack of nuclease enzyme. Now, where this total transcription sucker takes place, actually this total transcription takes place in nucleus. In nucleus, there is a nuclease enzyme there. If nuclease enzyme attack on the mRNA, there is degrade of mRNA. To prevent the degrade of mRNA, the 5-methyl cap formed here from the 5N and poly A also formed at the end of the mRNA. So now this is called as a post-transcriptional changes. Post-transcriptional changes. So after post-transcriptional changes, matured mRNA formed. What happened after matured mRNA formed? Actually, this mature mRNA formed in nucleus. Later, this mature mRNA translocated toward the cytoplasm. What happened when this mRNA translocated to cytoplasm? What happened in cytoplasm? Hello?
Translation process.
Right. Translation process takes place as a result formation of protein.
Proteins.
Now tomorrow class we will discuss the transcriptional changes. The clear about the splices, clear about the how the formation of 5-methyl cap, how the formation polyadenine. After that, eukaryotic translation. So far clear? Hello?
Sir, yes, sir. But one doubt, sir. Sir, in examination, we have to write both the allosteric model and torpedo model, sir, for termination.
Yes.
Okay, sir.
Both models. Even you put a headings. Actually, in our book, not given the headings. Just termination process there, but you have to write the termination 2 times, torpedo model and allosteric model. That much also enough.
Okay, sir.
Right.
These models are not there, sir, in the textbooks?
Textbooks not given.
Okay, sir. Sir, this
Right.
diagram we have to draw with this, sir.
Diagram no need, just explanation.
Very difficult are this diagram. Time will not sufficient for that.
Yes. But transcription diagram need. Transcription diagram, initiation, elongation, termination. And textbook given clearly. Page number 76.
Yes, 76.
What is initiation? Initiation attachment of the RNA polymerase to with help of the factors. That is initiation. What is elongation? The same RNA polymerase move forward direction downstream. And the same diagram, RNA polymerase end are separated from the stand. That is termination. Even take the picture from the Google.
Okay, sir.
Okay. Any doubt?
No,
Transcription, eukaryotic transcription.
no doubt, sir.
Please read once again.
Okay,
If you
sir.
have any doubt, ask tomorrow.
Okay, sir.
Okay. Can I quit the class?
Yes, sir.
Yes, sir. Thank you, sir.
Right. Thank you all.